And our first speaker is going to be Hongmei Li-Byarlay. I'm so sorry if I pronounced your last name wrong. She's an associate professor at Central State University, and the title of her talk is going to be Pesticide Residues in the Hive, in hive products and their potential risks to honeybees in China. So are you able to upload the presentation? Sweet.
Yeah, thank you.
Okay.
Yeah, thank you, Catherine. Can you see my slides?
Yep, we can see your slides.
Okay. Yeah. Hi, everyone. My name is Hongmei Li-Byarlay. I'm a research associate professor at Central State University at Wilberforce, Ohio. So this project, we have published it last year, and this is a collaboration with Dr. Luo's lab at the Institute for Apicultural Research of the Chinese Academy of Agriculture Research in Beijing, China. Honeybees are important pollinators together with other wild pollinators. Also, we know they have declined in their numbers and population and diversity in around the world in many countries. In today's conference, we are hearing a lot of talks on the pollinator decline. So pesticides as one of the main factors affect the pollinator decline. And a lot of the pesticides applied in the field on the crops and plants can get into the flowers, pollen, and nectar. And those are the main food for the foragers to go to collect. So the honeybee foragers can go collect the pollen, nectar with the pesticide residues and bring back to the hive. And in addition, with the beekeeping management practices, the colony can also have pesticides or medicides in the way from the management. So the pesticide residues are being reported from a lot of the publications before, and they're showing, a lot of them are showing not just really acute lethal effects right away, but instead it's a sublethal effect. We know now by a lot of publications, including our paper from 2019 paper, the pesticide residues can negatively affect the flyability, the homing ability, learning and memory, as well as other behavior of the honeybees. Sorry, I just remembered I probably need to set up a timer. I'm just gonna do a 10-minute timer. And so the plant we're focusing on for this project is the oilseed rape. Species name is Brassica napus. This is a popular plant in southern China, especially along the Yangtze River basin. So on the map at the bottom, you can see the main map of China. There's a brown line here. Can you see my cursor? I think. So
we can see your cursor.
Okay, good. Yeah, so there's a brown line. This is the Yangtze River from left Tibet all the way, go through the China land to the east into the ocean. So overall, we know there are more than 7.5 million hectares of oilseed rape in the area along the Yangtze River, and that's what I try to focus on. Another important reason to study this plant is because all these oilseed rape flowers bloom in the early springtime, and they are really good resources, almost the first type of food for the honeybees when they come out of winter. They will go to collect nectar and pollen from these flowers. And that is why these flowers are vital, and because they're really the first food that the bees can grab on when they come out of the winter. And so they really need these nutrition and nectar to build up their colony for the whole field season. However, there's not really much knowledge on the pesticide residues of the nectar and pollen of these honeybee colonies. So this is why we want to have the study. So the gap of knowledge here is the knowledge in what kind of pesticides and how much of these pesticide residues in the honeybee pollen and nectar. So for the bee bread, we actually have published another paper, but today I'm just going to focus on the pollen and nectar. For the sample collection, we have collected from 7 different provinces in China. So along the Yangtze River, you can see the red color pins and the purple color ones. So the red ones are the locations and field sites. We have collected only nectar samples. The purple sites are the ones we have collected both pollen and nectar. And on the right side is a really beautiful picture of the Yangtze River. To extract the pesticides, we use the GCMS, and for majority of the pesticides, so total 225 pesticides as the standards in the GCMS runs. For the LCMS, we use LCMS because we want to detect the more neonicotinoid pesticides using this method, which is more sensitive. How do we, so besides the toxicity of the each pesticide residue, how do we measure or evaluate the acute risks of these pesticide residues? We use the 2 major risk systems here. One is called a hazard quotient, the HQ. So this is the field application rate divided by the oral exposure, lethal dose 50, LD 50. And this is being referenced by Environmental Policy Committee and a lot of papers in general. Another database, another risk system is called a BRAX. This is referenced by the US EPA, Environmental Protection Agency. So the data we have collected include the toxicity of each pesticide, what we have detected from the GCMS and LCMS. We also calculate their hazard quotient of each pesticide. Now come to the, there are several slides of results. Sorry if you cannot see a lot of the small fonts here, but overall, in this table, we listed the compound of pesticides on the first column, and then the class of each pesticide residue. So they're either insecticide or herbicide for H, or the F for fungicides. And their total detection, how many samples out of the total 22 samples. And then the LOD is the limit of detection. Then on the right side here, the column is showing the pollen detection concentration rate. And just to highlight, so there are 3 pesticides here. They are phenylchalcone, phosmet, and then pyriproxyfen. Those are detected with a very high concentration. In the nectar samples, this is a similar table, and we have detected Binalaxia and pyriproxyfen, also in a very high concentration from these samples. So for the nectar sample, we have total 34 samples. This is a table, this is a graph to show you number of pesticides residues from different provinces. So Guizhou province, for example, from the 1 sample of pollen, we detect 1 to 9 numbers of pesticide residue. However, in the nectar, there are 9 samples detected pesticide residues, and 4 of them have about 9 residues, but another 5 samples, they have about 20 to 29 residues. So the highlight here is look at the high pesticide residues in the yellow bars. Those are the samples like from Hubei province, the pollen sample, and the Hebei and Yunnan province. These samples have more than 30, they have 30 to 39 of pesticides from those samples. So to compare a different year, because we have conducted the experiments in 2 years, both in 2017 and 18, we want to see if there's a difference in the year. And the results show there is a difference. We see a significant higher number of pollen, significant higher of pesticides in the pollen samples here. And then when we compare the nectar samples, we see the similar trend. So what that means here is that for some reason, I think in 2018, yeah, there are significantly higher number of pesticides applied onto these oilseed rape flower plants than 2017. So when we look at the hazard quotient, we detected 4 pesticide residues with the very high risks, including the carbofuran, cephalothrone, delta-matherin, and phenylchalthion. For the nectar, there's similar results here. These are the 4 pesticides. Some are overlapping with the pollen samples. So in the nectars, they are carbofuran, cephalothrone, delta-matherin, and fenprocatherin. So in a summary, we have detected 48 pesticides out of the 225, or more than 225 tested. So we detected 48 out of pollen samples and 34 chemicals out of nectars. And the maximum pollen hazard quotient values range very big, very widely from 0.16 to 700,000. You can see around that number. And the maximum nectar hazard quotient values are ranged from 0.07 to 185,000. So particularly because I think we use both GCMS and LCMS, we're able to detect a lot of several of these pesticides that are now reported before. So for example, the carbofuran, cephalothrone, delta-matherin, and phenylchalthion are relatively high with a high risks because they're HQ values. So definitely further testing is needed for these compounds. And overall, we think with this data, we're able to have a first really hand of data and knowledge now on the pesticide residue analysis. So yeah, I'd like to thank all the members at Dr. Luo's lab in Beijing, China. We had to work on this together, and the paper is published now. Thank you so much for your attention.
Excellent. I think just because we're at 3:14, we'll skip questions for now, but if you have any questions for Hongmei, I'm sure she'll appreciate if you maybe like send her a message, if that's all right.
Yeah, thank you.
Okay. Yeah. So I think now we'll just go on to the next person, which is